Okay, so we have gone over during this unit, this very feels like a very long unit. Enzymes, we've gone over the laws of thermodynamics. We went over photosynthesis, and now our last couple of lectures will be on cellular respiration, and then a little bit on fitness. So this will be 3 parts once again, because it's a little bit long, a little bit complex, but this will be the first part. So a review of our beautiful organelle, the mitochondria, right? This is our powerhouse of the cell, as everyone likes to call it. So just take a minute to review your notes and/or the text for the structures of the mitochondria, including the cristae, which are these essentially like the folds in the mitochondria, the matrix, the inner membrane, the outer membrane, all of that good stuff. Okay. So cellular respiration, this is how we essentially harvest chemical energy, which is stored in inorganic molecules like glucose, and use it to generate ATP or usable energy for the cell. All right, so we're going to have, this is our chemical equation for cellular respiration. So we have our organic molecules and oxygen as our reactants, and then we have CO2 and water and energy or ATP as our products. Okay, so starch is the major source of fuel for animals, and this will be broken down into glucose. And then we'll break that glucose down even further into usable energy or ATP. Okay. Okay, so going off of this, we are going to reintroduce the concept of redox reactions. So the catabolic breakdown of glucose includes redox reactions. Remember, oil rig or Leo Goesker, oxidizing is losing electrons, reduction is gaining electrons. So through this equation, you can see, for example, glucose is going to be oxidized into CO2. So it is going to lose electrons in this transfer of electrons, essentially. And then oxygen is going to be reduced into water, meaning it's going to be gaining electrons. So the oxidation of glucose transfers electrons to a lower energy state, and this is going to release energy to be used in ATP synthesis. So this is releasing energy in order to create ATP. Okay. All right, and then during cellular respiration, we're going to have the movement of electrons, just like we did during photosynthesis. So most of these electrons are going to follow a downhill exergonic path. So exergonic meaning they're going to be releasing energy, right? So glucose is going to, or electrons will move from glucose to this coenzyme called NADH. And we'll essentially be using NADH for the release of energy. We're harvesting energy from NADH. This is not the same thing as NADPH. Remember, NADPH is what is used in photosynthesis. This is not the same thing. So if you want to think NADPH has a P in it, so it's used for photosynthesis. That might help you remember the difference between the two, because these are not the same, even though they sound very similar. So electrons will move from NADH or NADH into another electron transport chain. So just like photosynthesis, cellular respiration also has an electron transport chain, which is just a series of membrane embedded proteins that electrons move through. And then they're going to move into oxygen. So oxygen is going to be the last electron acceptor, which we'll talk about more in depth later because it's very important for your AP exam. Okay, so just know that the last electron acceptor is oxygen in this pathway. Okay, so energy harvest. So glucose is going to be broken down in steps to harvest energy. So it's not just like, a one process and then we're done. It's going to be broken down in steps. So electrons are going to be taken from glucose at different steps. Each electron taken will travel with a proton. And dehydrogenases, and remember these are enzymes, ASE, they're going to take 2 electrons and 2 protons from glucose. So this is what's stripping away at glucose, essentially. What's breaking it down is these dehydrogenases, okay? These enzymes that take away 2 electrons and 2 protons from glucose. This is also the oxidizing agent for glucose. Oxidizing meaning we're going to be it's losing electrons, right? So that's where you see that here. So dehydrogenases takes electrons away and therefore is the oxidizing agent for glucose. Okay. This also transfers 2 electrons and 1 proton to the coenzyme NAD+, and that reduces it, right, because we're giving it electrons, meaning it's being reduced into NADH. So 2 electrons, 1 proton, meaning a hydrogen ion, are going to go into NAD+. That reduces it, makes it NADH because we're adding that hydrogen ion plus the 2 electrons. And this is what's going to store energy, and eventually we'll make ATP from this harvest of energy. All right, and then the other proton is going to be released in the surrounding solution as H+. So anytime you see that protons are being released into a solution, you should automatically think a gradient, right? So with the photosynthesis, we have the gradient in the lumen of the thylakoid membrane, and now we're doing almost the same thing. We're going to create a gradient of hydrogen ions, of protons essentially, that will then be used to create ATP. Okay, so this is a kind of an overview of what's going on. And then NADH will carry electrons to the electron transport chain. Right, so the electron transport chain is very, very similar, if not the same thing, essentially, as the one that we talked about during the light-dependent reactions of photosynthesis. So this is just a sequence of membrane proteins that shuttle electrons down a series of redox reactions, just meaning that they're literally going to be passing electrons to each other in this chain, right? So one's going to gain an electron and therefore be reduced, and then the other is going to lose that electron that it then gave to the next protein, and therefore it's going to be oxidized. So it's just a constant redox reaction for each of these as they are gaining and losing electrons. Okay, so this is going to release energy that's used to make ATP. And then the ETC will transfer electrons to oxygen. So remember I said oxygen will be the final electron acceptor. This will be to make water, but this is very, very important for the AP exam. I've seen so many questions that kind of hint toward this or allude toward this. So the final electron acceptor of cellular respiration is going to be oxygen. Okay, oxygen is the final electron acceptor. The ETC will transfer those electrons to oxygen, and it's the final one. So you might get questions asking, well, what if oxygen weren't present in the mitochondria? What would happen then? And you need to understand that because oxygen is the final electron acceptor, then there wouldn't be anything to accept that electron, and we'll kind of talk about what that means a little bit later when we get into it. All right, so this is going to release energy when the ETC transfers electrons to oxygen, and that'll be essentially, the reason for cellular respiration is to release that energy. So the stages of cellular respiration, there are 3. So the first one is glycolysis, and then we have pyruvate oxidation and the citric acid cycle. And then we have oxidate. I know this is a lot of like very large words, but I promise we'll move through them slowly and concisely. And then the last one is oxidative phosphorylation. So ETC and chemiosmosis is the last step in cellular respiration. Okay. All right, so we're going to talk about glycolysis, and then we'll talk about the rest of them in the next two lectures. But glycolysis is the starting point of cellular respiration. You can see by this image that it's going to happen in the cytosol. So this is before it gets into the mitochondria. And it's going to split glucose, which is a 6-carbon molecule, into 2 3-carbon molecules called pyruvate. Okay, so it's splitting a 6-carbon molecule, which is glucose, into 2 3-carbon molecules, which are pyruvates. Okay, and in your packet, I've given you this very complicated version of glycolysis. So this is the nitty gritty of glycolysis, but it is above or it is out of the scope of the AP exam, so you don't really need to know this for your AP exam, but if you are interested in it, feel free to look through it if you find this interesting or if you want to go into science as a career. This is a very interesting process. So if you want to look at that, it is in your packet, but by no means do you need to um, memorize all of these steps, okay? The summarized pathway, for those of you that just want to know what's going on, is that we have a couple of inputs. So we have ADP and an inorganic phosphate, and we have glucose. And our input is the ADP and inorganic phosphate, and then we also have 2 NAD plus or NAD plus. And then these are going to become 2 ATPs, 2 NADHs, and then 2 hydrogen ions, right? And then we're also creating water at the same time. So this is going to be the breakdown of glucose into 2 pyruvates. We start with ADP and inorganic phosphate, and then the 2 NAD pluses, and we end with 2 NADHs, 2 hydrogen ions, 2 ATPs, and then water. Okay, so those are our inputs and our outputs. Okay, and there's 2 stages of glycolysis. So we have the energy investment stage. So the cell is actually going to use ATP. It's going to invest ATP to phosphorylate compounds of glucose. So it is going to essentially kind of give up some ATP to then get back more ATP. So the next one then is the energy payoff stage. So this is when energy is produced by substrate level phosphorylation. So essentially, we're just using ATP, using that energy to then get more energy back in the later stages. So the net energy yield per 1 glucose will be 2 ATP molecules. So we get 2 ATP as a result of using a little bit of energy to get a little more energy back. Okay, and we also are going to get 2 NADHs. Remember, this is stored energy for the cell. This is going to be stored energy, and then we'll keep going and make more ATP in a little bit. All right, so again, so we have the energy investment stage of glycolysis. So we start with 2 ATP, and then we get to ADP and an inorganic phosphate. And then in the energy payoff stage, we're going to be using 4 ADPs and the inorganic phosphates to get 4 ATPs. Okay, so we get a little bit more energy back than we first invested. All right, so we have net 2 ATP, and then we're also during this energy payoff stage, we're using 2 NAD pluses. Remember, that's that coenzyme, 4 electrons, 4 hydrogens to then create 2 NADHs and the 2 hydrogen ions. Remember, these 2 hydrogen ions are going to then be released into into the matrix essentially, and it's gonna cause a gradient. All right, so the net yield essentially will be 2 pyruvates and 2 molecules of water, and then 2 ATP and 2 NADPH, or sorry, I just said NADPH, not NADPH, 2 NADHs or NADHs, and 2 hydrogen ions. Okay, quick check. Where does glycolysis occur? So write that down real quick. Where does glycolysis occur? And you were right if you said in the cytosol. The next one is what is produced through glycolysis? And you would be correct if you said pyruvate, right? So through glycolysis, we're breaking glucose down into 2 pyruvate molecules. And then what is the net production of ATP during glycolysis? And you should have said 2 ATPs. Great. And that is it for this one. Have a great day. I'll see you guys tomorrow.